Oh. এই প্রতিমা ওকে একটু বলো না একটু এখন চুপ করতে। এই বন্ধুরা একটু সমাজে বলো না ওয়াজ কি আর এখন ওয়াজ করছি না এখন কি বলবো। Okay. So this is a Malpighian tubule which is present in all insects. You have the germarium. Here the egg development starts to take place. Then you have an egg chamber. Then the follicle cells are formed. This is how a mature oocyte is formed. So this is how the development takes place. And in the development, as you can see in this picture, this is the fertilized egg. And this single egg divides. And then the division takes place without the formation of, so division of nuclei only. Not cytoplasm. This is known as syncytium. So you have this single egg. Like I said, the nucleus divides many, it goes to the periphery, and you have these cellular blastoderm formed, which they are basically the follicular cells, which you had seen in this picture over here, the follicle cells which are forming over here. And this is a mature egg. This is the egg which you will see deposited by cockroaches, but it happens the same for Drosophila also, all insects. Now, let us, I'll close this and we will go to the main study. Are you able to see this? Those who are online?
Yes, sir.
Yes, sir.
Okay. So essentially, there are some group of genes which are responsible to give anterior, posterior axis. Okay. So if this is an egg, which will be the anterior, where will be the head and where will be the back? So that this is the anterior side and posterior side. So you have an egg. So this anterior side and posterior side, how this axis is formed. If you look at this, how this axis is formed in the anterior portion. This is an axis. And this is the dorsal and this is the ventral side. If I go up, I draw this egg over here. This is the dorsal side and ventral side. and this is the anterior side, and this is the posterior side, and this is the axis, anterior posterior axis, which is front and which is back, anterior or posterior, and এটা হচ্ছে পিঠের পোর্শন, এটা হচ্ছে তলার পেটের দিকে. How this region is determined? So this is basically determined by set of genes. There are 4 set of genes which do this. The first set is known as the maternal effect genes. Then you have a set of genes known as gap genes. Then you have the pair rule genes. Then you have the segment polarity genes. Along with them, there you have got another set of genes known as homeotic genes. So far, so clear. 4 sets of genes are present. First is the maternal effect genes, then the gap genes, pair rule genes, and segment polarity genes. You people are able to listen to শুনতে পাচ্ছো?
Yes, sir. বুঝতে পারছি।
ওকে, একটু মাঝে
হ্যাঁ
মাঝে বলো, আমি বুঝতে পারছি না, আজকে খুব ডিস্টার্ব হয়ে যাচ্ছি আমি। ওকে, so as I said, this anterior posterior polarity of the embryo, larva, and adult, it has in the polarity of the egg. So which will be the mouth region and which will be the anus region, that is determined just where when the drosophila is in the stage of the egg. So in the mother's ovary, a group of genes are expressed and they are known as the maternal effect genes. Okay, so what they do inside this ovary, here the egg is over here, they keep on injecting or some gene products. So these maternal effect genes are expressed in the ovary of the mother, and then this gene product, what happens, the messenger RNA, they diffuse into the egg. Not only they diffuse into the egg, they go diffuse into different regions of the egg. Say the first product is in the anterior end and the second product is in the posterior end. It can be something like that. Now, and these products are placed in different regions of the egg. So maybe product 1 is present over here, product 2 is present over here. Okay, and now these messages, you have read this clear before about the gene which is the DNA. They produce messenger RNA, central dogma. Okay, these are the by the process of transcription, and they will produce the protein by way of translation. So these maternal effect genes are present in the mother's ovary, that is in the form of DNA. Then transcription takes place. Transcription takes place and mRNA is formed. Now these mRNA diffuse into different regions of the egg. Clear? Yes,
Yes,
no?
sir. Yes, sir.
Yes, sir.
ওকে, এটা খুব সুন্দর ইয়েস বললো, শুনতে পেয়েছি। Now we go back. So these messages are nothing but the mRNA. They encode transcriptional and translational regulatory protein that diffuse through the syncytial blastoderm and activate or repress the expression of certain zygotic genes. So they enter into the egg, and after it enters into the egg, they are responsible for activation or for repression of certain uh, genes which are present inside the egg, not in the mother's ovary. So the maternal effect genes, they are expressed in the mother's ovary. If you look at the picture over here, cytoplasmic polarity, this will be the red, maybe the anterior, and this is the posterior. So this is by effect of these maternal effect genes. So where they will go inside the egg, that side will become either anterior or posterior depending upon which product it is. So two of these proteins which enter after transcription and translation, the name is bicoid and hunchback. They are responsible for the formation of anterior structure, the structures which are present in the anterior end. And nanos and caudal, they regulate the formation of the posterior parts of the embryo. So far clear? Yes, no, Bolo?
Yes, sir.
Okay. Now let us go into a bit of a details on how this thing takes place. So we have seen that bicoid and hunchback mRNAs, its protein products are critical for the formation of head and thorax. Insect has got three parts: the head, thorax, and abdomen. So bicoid and hunchback messenger RNA, they are maternal effect genes from the bicoid gene and hunchback gene produced in the mother's ovary. This messenger RNA, his protein products, bicoid and hunchback, they are needed for the formation of head and thorax. If these genes are mutated, then in this fly, the head and thorax do not form. They don't survive actually. But you can see that embryo, that the head and thorax have not formed. Apart from that, you have the nanos and caudal, whose mRNAs, whose protein products, they are critical for the formation of abdominal segments. So the abdominal segment formation, you need two more maternal effect genes, that is nanos and caudal. Okay. Now, if you look in the oocyte mRNA, the egg, you will see in the anterior region, the concentration of bicoid mRNA is much more higher. And in the posterior region, এই দেখো উপর থেকে যত এই দিক থেকে মানে এইটা, this is the egg. The concentration of bicoid is, this is bicoid mRNA here. This is much more higher over here. ঠিক আছে, অনেক বেশি থাকে যত anterior, maximum in anterior. There is a gradient. Concentration falls. In the anterior end, this is the maximum, and somewhere in the middle, this is the minimum. And in the posterior side, there is no bicoid mRNA. Similarly, মানে দেখো লেভেলটা, red level from high to low. As it goes from anterior to posterior, the mRNA levels, they decrease. Similarly, nanos mRNA is there. It is high in the posterior side. and as it goes towards the anterior, its levels decrease. ঠিক আছে? That is because bicoid mRNA gets translated to form the bicoid protein. And okay, one more thing, remember that the levels of the mRNA, not the protein, mRNA level of hunchback and caudal, they are the same. Fine. mRNA levels of hunchback and caudal, they are the same. mRNA, not the protein. The protein is actually the functional product. Now what happens, this caudal mRNA needs to be to the process of translation. It forms the caudal protein. Now when bicoid mRNA is translated, this is the process of translation. It forms the bicoid protein. It inhibits the translation of caudal mRNA to caudal protein. So if you see the level of caudal protein, just because bicoid is high over here in the anterior region, this caudal protein is not made because bicoid protein stops the translation of caudal protein. Clear? Yes, no?
আরেকবার রিপিট করবেন স্যার?
হ্যাঁ
স্যার আরেকবার রিপিট করবেন লাস্ট
আরেকবার
পয়েন্টটা?
রাইট রাইট লেট মি রিপিট দিস ইন এটাতে ভালো হবে আবার এসে গেছে this is the egg, this is the anterior region, say, and this is the posterior region. Now anterior region, I mean I'm in the anterior region, high concentration of bicoid mRNA. And this is in the posterior region, high concentration of nanos. mRNA. Now mRNA levels of caudal and hunchback are same throughout the egg. So far clear? Yes, no?
Yes sir, clear.
Yes, sir.
Now, so এখানে there are lot of bicoid mRNA এই দিকটাতে maximum. These are bicoid mRNA. যত এদিকে আসছে কমে যাচ্ছে। ঠিক আছে? এদিকটাতে in the anterior end bicoid mRNA, a lot of bicoid mRNA is there. Gradually their levels are low. In the posterior end you have high levels of nanos. এই যে আস্তে আস্তে কমছে। This is the high level of nanos mRNA. Now bicoid black color দিয়ে দিলে বেটার হতো। Bicoid mRNA It will form bicoid protein. Clear? And
Yes,
we
sir.
know that
Yes, sir.
bicoid mRNA will translate and form bicoid protein. It is clear.
Yes, sir. Clear.
Yes, sir.
Okay. So in this region, lots of bicoid is there. So more bicoid protein will form here. And in this region, more nanos protein mRNA is there. So more nanos protein will form. So far, this is clear.
Yes, sir. Clear. This is clear.
And a gradient is formed. At a bicoided nano, the gradient is formed. This is the gradient of, this is bicoid, and a gradient of nanos is formed. Clear? The more anterior, more nanos. The more posterior, more nanos. Less posterior, less nanos. It's like a gradient. Gradient means ki on one end you have high, then gradually as you go to the center, the concentration decreases till there is nothing. A gradient, that is the meaning of a gradient. Now, bicoid protein, what it does, it stops translation of caudal. So in the, now we have seen that the mRNA levels of caudal uniform. Uniform means it is same throughout the egg. Not like bicoids or bicoid and nanos, where the bicoid is concentrated on anterior end and caudal is lot of caudal is present in the posterior end, and it exists as both of them exist as a gradient, highest in the end and lowest in the center. Caudal is same. So at the anterior end, when bicoid is stopping the translation of caudal, what happens? No caudal protein in anterior end. Clear? Yes or no?
Yes, sir.
Yes,
Clear.
sir.
No, because there is lot of bicoid protein. They're stopping caudal translation. Although messenger mRNA is uniform, these messenger mRNA of caudal which are at the anterior end, they are blocked. Their translation is blocked. Clear? Okay. So there is no caudal in the anterior. But in the posterior end, there is no bicoid. So caudal protein is formed in the posterior end. Is formed in posterior side. So nanos protein plus caudal protein, we have seen they are responsible formation of abdomen. So already now, depending upon where bicoid concentration is high or nanos concentration high or caudal concentration high, already a polarity is formed. You know which side is going to form the head and which side is going to form the abdomen. The polarity has been made, established. Similarly, just the other thing which happens, you have nanos protein it inhibits the translation of hunchback. So concentration of hunchback is uniform throughout the egg, but nanos is inhibiting. So nanos will not be expressed in the posterior end. Hunchback will not be expressed in the posterior end. Like caudal is not expressed in the anterior end. Nanos is not present in the anterior end of the egg. So there you have hunchback protein is formed. Hunchback means in this drosophila, the mutants where this hunchback gene is mutated, the back is somewhat elevated like that, like a hunch. So these are very important genes. Any defect in these genes, it is lethal to the fly. So these hunchback genes, and you know now that the hunchback plus bicoid, the protein product, are needed for formation of head and thorax. Is this clear? Yes, no?
Yes, sir. Clear, sir.
Okay, এবারে বুঝতে পেরেছো তো? তাহলে very good.
Yes.
এটা একটু now you have come to the complex chapters. Now এবারে simple thing happens. So this matter, now we are moving into the egg. We have moved because the maternal effect genes, bicoid and nanos, they have come inside the egg and become protein. And next set of genes which are expressed by them, they are known as zygotic genes. এদের বলা হয় zygotic genes. So we know that bicoid is in the anterior end, nanos and caudal at the posterior end, bicoid and hunchback. Now they will activate or repress a group of genes known as zygotic genes. These zygotic genes, they are regulated by these maternal factors, and they are expressed in certain broad segments, about 3 segments wide. If you look at a stain these mRNA, you will see that roughly there are 3 broad segments, 1, 2, 3. So there are different zygotic genes. We will know the names. Example, Kruppel, Kruppel. And these genes are known as also gap genes. Zygotic genes are also known as gap genes. Because if one of these gap gene is mutated, you will find huge segments of the fly missing when it develops. Of the embryo, huge segment, because that gap gene was not expressed. A huge gap is formed. Example is Kruppel gene. Now, gap genes are transcribed and translated. Now again, like you had seen, in bicoid and nanos, you have differing concentration, more in the anterior side of bicoids and less in the posterior side. Similarly, these gap genes also form a gradient. So, differing concentration of gap gene proteins, which is very high, say for example, one gap gene group, Kruppel, is very high near the 4th parasegment or 4th segment. And at the end of the 4th segment, the concentration of this Kruppel is very low. So the effect of Kruppel will not be there. So the genes which Kruppel repress, they will be expressed in the posterior side, not on the anterior side. Same what happens to bicoid and caudal. And because of this, instead of bicoid and caudal, what happens? You have here these gap genes will express another group of genes known as pair rule genes. Example of this pair rule gene is Fushitarazu. The name of this gene is Fushitarazu. Actually, the transcription of the different pair rule gene, it results in a striped pattern of 7 vertical bands. If you look at this picture, 7 vertical bands are formed. 1, 2, 3, 4, 5, 6, 7. They will again be divided into 14. Okay. Now, after this comes the segment polarity genes. So already you have a egg. So pair rule gene activate the transcription of segment polarity genes, whose mRNA and protein product divide the embryo into 14 segments. They are known as parasegments. So 14 segments are formed. In the end, so gap gene, pair rule gene, and segment polarity gene, the 3 of them together, they are also known as the segmentation gene. So they include the gap gene. Gap gene ke zygotic, they are all zygotic gene because they are formed in the zygote. Gap gene, pair rule gene, and segment polarity gene. Now, in this segment polarity gene, these parasegment segments are formed. So you have, say for example, thoracic segment 1, the first thoracic segment. They also divide it into anterior and posterior side. Even each segment has got an anterior and a posterior. Right? So they give different segments. So it is divided into 14 parasegments. Each segment will give rise to, say, for example, in the thoracic segments will give rise to legs. At the same time, when the segment polarity gene works, another group of gene known as homeotic genes are expressed. Homeotic selector genes. Their transcription, when these homeotic genes are expressed, determines the developmental fate of each segment. There is a the homeotic gene known as antennopedia. So, in each segment, this antennopedia gene is expressed. But when it is mutated, an antenna is near in the head region, an antenna is needed to be formed. But instead of that antenna, you have a leg coming out. That is because it's a mutation. Similarly, where the leg is supposed to come out in the thorax, you have legs. This antennopedia is repressed. But when it is repressed in the head region, or a mutation is there, and you have the another leg coming out over there instead of the antenna. What happens in the thorax? Opposite happens in the head region. If the same happens, you have legs coming out from the antenna. And these flies are viable. You have seen them without antenna. They don't survive much because they can't feel much because you have legs over there. But these are controlled by homeotic genes. So this is how actually maternal effect genes or the anterior posterior axis formation takes place in Drosophila. Hope I'm clear. I'll stop over here because it will be overdose if I start dorsoventral polarity. But read this from this note. I don't know if I've sent it, but I'll send it today. I'll send this notes. আচ্ছা, কোয়েশ্চেন্স?
নো স্যার।
Yes, sir.
কোনো কোয়েশ্চেন্স আছে?
Yes, sir.
হ্যাঁ বলো।
স্যার এক্সামের ডেট তো শুনছি মানে এবার দেবে দেবে করছে অফলাইনের, কখন এত পড়বো স্যার? এটাই
এক্সাম
কোশ্চেন।
এন্ড অফ বোধহয় জুলাই টুলাইতে হবে, জুনটা পুরো পাবে তোমরা।
আচ্ছা স্যার।
ওকে, আই এম স্টপিং দ্য রেকর্ডিং।
ওকে স্যার, থ্যাংক ইউ।
থ্যাংক ইউ স্যার।
ইউ ওয়েলকাম।